As soon as, the structure of DNA was resolved, and that people started developing molecular tools to read the DNA, the famous works of Sanger, who invented sequencing, and also then the need maybe to do all the genetic manipulations. So very early on, people started, reading and synthesizing DNA, and so then if you synthesize DNA, you can also encode some information.
This is Christophe Dessimoz, professor at University of Lausanne and at University College London.
So they were very small bits of information, for instance, some secret messages, like, 1944 invasion of Normandy. Someone wrote that in DNA, or stored some little poems and drawings in DNA. But these early attempts were very laborious, and that required a lot of ad hoc steps, and at the end of the day required also ad hoc encoding schemes.
But things have recently changed.
The revolution has been in the recent years with a massive decrease in cost of sequencing. So now it's very, very cheap to read DNA. In fact, every molecular biology lab these days reads DNA all the time. Actually, DNA or that comes from the genome, or DNA that was first expressed in form of RNA and that is translated back into DNA for the sequencing. And this is a very kind of a routine technique in molecular biology. And as a result, people, such as our team a few years ago and others, have thought, okay, if we could read this so efficiently, and in a way we know also how to write it, perhaps we can use it to store arbitrary information.
Basically, we can now easily read DNA. We are halfway there. To make DNA storage a reality, we now need to efficiently write DNA.
The synthesis costs remain relatively expensive. So there's a huge market force to try to read DNA, right? This I mentioned, this is the basic technique now in molecular biology. All life has some DNA, and so it's extremely interesting to understand this diversity for, let's say, fundamental biological reasons, but also for very applied, diagnostics, like discovering some outbreaks or understanding the cancer in particular individuals. And so there's a big market force to drive to sequence the DNA. But to synthesize it, actually, although a lot of people need short bits of DNA, for instance, to amplify some specific part of the genome, there was just not, I think, as much demand for long genes.
But why is writing so much harder than reading?
There is some trends now in synthetic biology to try to synthesize entire genes, and there are some market pressure maybe to do a little bit more of this. But I think it's nowhere near what we've seen for the reading.
Now, that does not mean that there is no research in DNA synthesizing.
We need new type of applications where that will drive the cost lower, and quite possibly storing information in DNA is going to be that application. Certainly, in the last few years, we've seen a huge interest and fascination also from not only from the public at large, but also the IT industry specifically. For instance, Microsoft last year stored an entire movie in DNA, and there's a number of other companies that are investing in this space.
Do researchers need a major breakthrough? What is the main technological hurdle to DNA synthesizing?
I think there is a need for maybe, a bit of patience, which is easy to do. I'll come back in a few years and ask me the question. But I think I think incremental technological progress can get us there. And what we need also is just like the incentives to do that. And so I think but I should say it's a very, very young field, right? I mean, this pioneering work, based on next generation sequencing, they were published in 2013. I mean, we were not the only ones. And since then, there's been an explosion also of contribution. And, these 4 years, it's a very little time even, to set up companies and start doing some development. And so, I think the main challenge at the moment is costs, but I think the costs are going to go down, and I don't think we need really a technological breakthrough. I think we need, some optimization and then, getting at things at scale, but it's just a question of market force. And by the way, as long as you don't have like a market in place, it may be difficult for people to invest because, sometimes the timing is very important with these things. So, and it's not necessarily, if you're the first one that you are going to end up like having the entire market. After all, DNA is the molecule of life. I mean, there's, there are probably some efforts to manufacture, to use other sort of very small molecules to store information. Maybe, there was work in the 80s to synthesize a non-natural amino acid. There are some non-natural also nucleotides. So, conceivably some companies may look into these type of technology, perhaps to also patent the work and make sure that they can that they can protect their investments. But yeah, I mean, this is all like a business speculation. I'm not really poorly qualified to talk about this. Perhaps you should go and talk to some colleagues we have in the other faculty here.
But with the technology still in the early stages, converting digital bits into DNA code remains both slow and expensive.
We can find some DNA that is tens of thousands or even hundreds of thousands of years old,